Hello, everyone. I'm Kevin McKernan from Medicinal Genomics. I've got about a 30-year experience in the genomics field. I've built DNA sequencers, and I make PCR assays all day long. So I applied some of that background to looking at how much DNA is in these vaccines. And by now, I think many people in the audience know that about 10 studies out there have found DNA contamination in these vaccines that's either right at the limit or above it. And the numbers vary quite a bit. So Jessica, Charles Rixey, and I set out to figure out why is everyone getting different results. And what this paper shows is a mechanism for why these mistakes are happening. There is what's going on in the regulating of these vaccines is they're looking at a tiny 100 base pair region of the plasmid that could contaminate these vaccines and using that as a proxy for the rest of the plasmid and how much might be there. And that mathematical extrapolation is fraught with a lot of errors, and that it makes the assumption that all parts of the plasmid are readily degraded at the same rate. And what this paper identified is that that's not true. The spike region of the plasmid does not get destroyed with the methods that they're using. They're using an enzyme that's incapable of erasing RNA DNA hybrids. So when they make these vaccines, they make some RNA off of the DNA, and that RNA is stuck to the DNA, and the enzymes can't erase that RNA DNA hybrid. There are other enzymes that can do this, and we show this in the paper, that if you use the appropriate enzyme, you can erase the spike DNA as well. But this really highlights the mechanism of action and why regulators think this is below the limit and everyone else finds it above the limit, is everyone else are using tools that more comprehensively measure all of the DNA that could be there as opposed to a really small target.
It was basically the result of 2 days of lab work where we pretty much formally demonstrated why we think that most of the DNA that are in the Pfizer and Moderna vials are there. And it comes down to improper enzyme use during the cleaning phase, which is, just to reiterate for everybody, these nucleoside modified mRNA lipid nanoparticle encapsulated products are completely different from conventional vaccines in that they have this, this mRNA component, which is modified specifically. And as part of manufacturing those nucleoside modified mRNAs, they have to go through a specific production process, which when it was upscaled, they used a plasmid E. coli system. So it started from DNA, in vitro transcription was done, and that got them to the nucleoside modified mRNA part. Now, the problem during that process is absolutely definitively at the end, when you have all that stuff left over, the residuals and the contaminants, you have to clean up the end product, which is supposed to be your nucleoside modified mRNA. And so what they traditionally do is they use an enzyme called DNase 1. And as Kevin pointed out, DNase 1 is not effective at clearing out DNA RNA hybrids, which absolutely naturally form. I have a feeling that since we're able to show this mechanism of action of a lack of functionality of the right enzyme, that people did know about this. I would find it very hard to believe that they didn't. The other thing is, if the enzyme, if this specific enzyme DNase 1 was ineffective at removing DNA, and they actually, did what they were meant to do, and follow up and test the level of DNA at the end to make sure that they had a pure product, they naturally would have seen relatively high levels of DNA, which they did. We have documentation to show that. However, they threw that under the rug by saying that it came under some, European Medicines Agency threshold, which is irrelevant, again, because that's based on naked DNA, not lipid nanoparticle encapsulated DNA. So my suspicion is that maybe, I think Kevin's right. I don't think anybody knew the implications of what they were doing from a human physiology point of view and a destructiveness point of view, but I would find it hard to believe that somebody along the lines didn't realize that this specific problem was a specific problem.
I'll add in on that, Jessica, and that's our paper does link to a citation, two important citations. One is from Sutton et al. That demonstrates this enzyme can't process what they're processing. The RNA DNA hybrids cannot be processed by DNase 1. Sutton et al. points that out. There's another one from Lemp et al. That is actually from BioNTech themselves publishing that they know about this, that this enzyme is 100-fold less effective at erasing DNA that's hybridized to RNA. So it's known. This is something that they certainly had the assays to know was occurring, because you can look in the EMA documentation and see that they have a spike encoded qPCR assay, and they're not reporting the CT values for that, but they ran it to prove that the plasmid insert was there. So they have a CT value from Spike and they have one from CAN, and I bet those don't agree, just like ours don't agree. They're about 6 to 7 CTs off, which is about a 100-fold difference. So this 100-fold difference is why the regulators think it's fine, because they're getting numbers from the part of the plasmid that can be erased with DNase I, but they're not bothering to look at the other parts of the plasmid that are not getting erased by this enzyme. And that's why everyone else is seeing 100-fold more. So it is a real problem. And the call to action here is pull these until they clean up this manufacturing, because there are papers linking excess DNA to myocarditis. When you have excess DNA like this, it triggers a pathway known as C-gas sting, and that is known to be activated during myocarditis. We also have papers listed in there that demonstrate this can trigger oncogenesis. If you chronically stimulate C-gas sting, it can lead to oncogenesis. So Quan et al. Describes that. So call to action here is pull these until the manufacturing is actually clean, and they can meet the guidelines. And the second call to action is for the community to come together and figure out what is a tolerable amount of DNA in these vaccines. We're in a world now where we can sequence every nucleotide in these vaccines. When these limits were made, we did not have next generation sequencing. We were guessing what DNA could do if you put it into rats. Now we can actually read genomes and figure this out. So small amounts of, 10 nanograms of DNA, this limit really came about due to thinking about host cell contamination. 10 nanograms of, let's say, chicken DNA or human DNA that might be in a background of another vaccine is only 1,400 copies of a genome, but 10 nanograms of a plasmid, that's about 1 billion copies. All right, so it's really not the weight of the DNA that matters, it's the copy number. That's what we pay attention to. In fact, Keith Pettin's work at the FDA points this out, that if the contaminating DNA is a viral element or a plasmid, the limits need to go down 1 millionfold, because that is much more problematic based on its copy number. So we have to, this 10 nanogram limit, I call it's a limited hangout. Everyone's talking about it, but it's irrelevant right now. What really matters is we have the tools to measure all the DNA in these vials, and we should have a very thorough discussion about the nature of that DNA. Random DNA is one thing. Gene therapy plasmids that can integrate, that bind to p53 is a very different beast. And we shouldn't be tolerating that much DNA in these vaccines.
Yeah, that's exactly what I was going to say. I mean, I don't think any amount of DNA in this specific context is safe. I think the platform is just, it's never going to work. So I think the moratorium would last for a lifetime. But that's exactly right. I mean, we're not just talking about any random DNA. And I'm not sure everyone's circulating the literature world, but there's a famous and infamous cancer researcher who's really high up at Brown named Wafik El-Dari, and he's recently published an article on the emergence and the emergence of raging cancers that's happening all over the world. I mean, there's no one you can turn to and not hear a cancer story from, and it's getting cumulative. Like the rates are going up of rare cancers, aggressive cancers, and this is linked potentially to the DNA story because of this SV40 element, the promoter enhancer, which Kevin said. When this is published, it binds to p53, which is the guardian of the genome, and there's just never going to be any good outcome from binding an essential element in tumor suppression. So it's not the only thing that it does, too. I mean, it's the base question is, what is this gene therapy plasmid doing in this context in the first place? I mean, it's SV40 didn't really have a role as a promoter.
It's superfluous. They don't need it there. Moderna doesn't have it. So that's exhibit A, that you don't need this there. It does raise further scrutiny on this limit, because this plasmid can self-replicate in 1 million cells. So you could sneak one molecule in there and it'll make more once it gets in.
That's really exactly right.
Can drive a truck through that regulation.
Yeah, there's no safe limit in my opinion. But yeah, and again, back to the whether or not any of this was known or if this was just ignorance. I mean, we brought it to the attention of Canadian health regulators at Health Canada for primarily that this was a problem. And I've written about this many times. One of the regulators infamously said that SV40 is not functional, and it's just it's a ridiculous way to respond to a potential crisis. Actually, I don't know how else to define it. Because you have no data to support that claim, first of all, and there's no reason to believe that it's not functional because it is functional.
Yeah, there's lots of literature showing it is. There
Yeah,
isn't any literature showing it's safe.
yep. It has absolute functional roles in gene therapy as trafficking other molecules to the nucleus of cells. And then you have to start wondering beyond the fact that any foreign DNA, when presented to the cytosol of a cell, can induce cancer pathways, are we looking at potential integration events? Like specific or non-specific. I mean, either way, it could cause damage. And it could cause generational damage if you're talking about germline cells or stem cells. So yeah, it's not something you can just sweep under the carpet and say, oh, it's under this limit that's irrelevant, or oh, it's not functional. Like without even like the thing about this is like I said, this took about 2 days to do this work, which was fantastically fun and illuminating. Um, we're independent scientists. We're not getting paid to do this. Actually, there was a lot of money spent to do this on our own time. So it's funny to me that the vested interested people, the regulators, the manufacturers, whoever you want to point to, they had to have looked at this either before or after we raised this issue with the hybrids. I mean, because it's like Kevin said, it's published. There are enzymes that are associated with being able to degrade these guys and not. And by the way, we do have enzymes in our cells. It's called or RNase H, sorry, that can degrade RNA DNA hybrids by nature because they do form naturally. But you don't want to impose excessive amounts of exogenous foreign RNA DNA hybrids to a cell because then you're just going to uh, you're going to present too much mass of a problem and a substance for the cell to be able to handle naturally. So that's another actually good point because, and this is all published as well, when these RNA DNA hybrids form, they create these things called R loops. Kevin can elaborate. And these actually like lead to disease states. And one of those disease states is cancer if there's an overaccumulation of these R loops. And that can happen if you have an RNase H deficiency. This is published. Or if you, like I said, exogenously introduce any old cell with hordes of foreign, RNA DNA with these R loops.
Yeah, it will trigger different toll receptors when it comes in through the cytosol. Typically, those R loops are being found in the nucleus, and the RNase H is doing its work in there. But once you introduce these in the cytosol, all types of other pathways get triggered because I think there's some type of viral infection going on. And so
Right.
it's not something you want to flood the cells with. There's one other detail that is, I think, important to put in context here. And Retzef Levy, who's on the ACIP committee, published this work with Josh Gitzkow, which is when they originally made these vaccines in the clinical trial, they didn't use this process. And that is, I love that. Yeah.
Isn't that great?
That's an important detail because what they did in the trials is they generated this DNA with PCR, and they used that to make the RNA. And yes, RNA DNA hybrids happen in that process as well. But that DNA is arguably easier to get rid of because they could attach a biotin to the DNA and fish hook it out with a magnetic bead, and it could be a cleaner process that way. What happened after the trial, which is published in Retzef's work, is they switched to a whole new manufacturing process that put this stuff through a coli and included all these plasmids. That does several things. One, there's now all the SV40 components are now in there at the same copy number as the spike. But more importantly, when you run this DNA through E. coli starts to methylate the DNA, and it methylates the A's, and that's not natural for human DNA. Human cells don't like to see methyl A DNA in the cytosol. They think there's a virus or a bacteria, and it starts triggering like a sepsis response. So this change in manufacturing is very material, and it's important for people to know that the vaccines that are on the market never went through a clinical trial. They're
Hmm.
different. They're biologically different. They're methylated differently. They have different types of DNA present. And there's probably some contaminants from the E. coli process as well that we don't have great tools to measure right now. There's something known as endotoxin that comes through that process that has been redacted in most of the Pfizer documents. So we don't know how much of that's around. But that material change from process one to process two was published in the BMJ. And the world should know that the vaccines they're taking have zero clinical trial data, none. Even if Brooke Jackson's done a great job showing all the problems in the initial clinical trial, but we're not even talking about that material. This is something completely different that has no trial data, and it has known contaminants now that weren't in the clinical trial that we have linked to myocarditis and cancer. So this methylation problem is a second issue that are in the process. When Jess and Charles were there, we actually sequenced all of these things on Oxford Nanopore, and we could look at the methylation profiles. It's methyl A is everywhere, and there's another methyl C region that's getting methylated from the E. coli strain they used. And that's another mistake they made. There are E. coli strains that don't do that, and they didn't use them. And that's really
That's right.
bizarre because most plasmid manufacturing for pharmaceutical companies make sure to use E. coli strains that don't methylate the DNA like this because they know that methylated DNA is hyperstimulatory to the C-gas sting pathway.
And they would have known that. Like I had to ask Kevin like three different times. Are you sure they would have known that
Yeah,
they
yeah. There's
should
a
use
paper I
specific?
have. We've listed in this paper showing that it's from 2010 about plasmid manufacturing for transfectable reagents like this. You have to use a DAM DCM minus a knockout strain in E. coli to make sure you don't have inflammatory DNA in there.
And Kevin's not cursing. It's actually called damn DCM.
Sorry, it stands for DNA adenosine methylation. It's an acronym.
That's actually yeah, it's something that everybody, if you don't know already, is what I was alluding to before, which is this bait and switch so-called. So yeah, there's no way that you couldn't anticipate the problems that we're actually seeing a lot from upscaling to this E. coli plasmid system with all of the things that they did wrong. Um, and it would have been easy to see, like all they had to do was all the things they always did at the end of the manufacturing process to see if their product was so-called pure. And that would have meant that it was only nucleoside modified mRNA, which it
Yeah.
obviously wasn't. So we have to assume, in addition to everything that we found, every time Kevin turns over a rock, from a genomics point of view, or from a sequencing point of view, or from a QP, like every point of view, we seem to find something new. So again, this LPS, the lipopolysaccharide problem that might also be causing anaphylactic reactions, because there have been a lot of those reported in VAERS and other pharmacovigilance databases as well. I have no doubt that there's lipopolysaccharide in these as a byproduct of using E. coli. Because if they didn't, it's just logic, right? Um, if they weren't successful or they didn't do due diligence and clean their product at the end for one thing, maybe I'm wrong. Maybe they did just only use the wrong enzyme, and maybe there isn't like polysaccharide, but I would assume that there is in some of the vials. And
I
yeah, they
think
didn't test it on anyone.
Yeah, an important point here is, as Jess mentioned earlier, this is about 2 days of work in the laboratory. So this is not rocket science. I know we're talking about DNA and it sounds very complicated, but the tools we use to measure this, the qubit fluorometer is around probably $5,000. And in 2 days, Charles Rixey, who's a veteran, he came in on Veterans Day for this, it was able to sequence the vaccine on an Oxford system, and he found a 5.2 kb, a 5,000 base pair read a fragment in these vaccines. So when people tell you that, oh, it's all fragmented, don't worry about it. No, we have entire coding regions in these vaccines, like molecules that are so long that they encode the entire spike region, and the spike DNA of this vaccine has a promoter in front of it that's active in 1 million cells. So if that piece of
Yep.
DNA gets in, it's going to turn into spike RNA and it will express. So this helps explain potentially why we're seeing patients out there that are still expressing spike years later. Nurse Lindsay is one that's been documented, maybe 2,000 days, she's still expressing spike. The only way that makes sense is if these vaccines hit some sort of immunoprivileged stem cell that doesn't get cleared out, and this DNA is sticking around and continuing to make spike RNA inside those patients years later. That was not part of the informed consent. The informed consent was it stays in the arm and it's gone in 48 hours. We now have papers showing it's out to 17 months, patients that have spike expressing for 2 years, and it's all over the body. So the biodistribution's wrong, the duration and permanence are a problem, and the contamination that we're finding in here is likely the cause for this. And we know the mechanism of action. We've zeroed in on what part of the pharmaceutical process is doing this. It's very easy to fix. I think I'm a little bit more hopeful. I think they can get rid of all this DNA if they use the right enzymes. I'm not hopeful they're actually going to be forced to switch because there's no liability for them. Why would they?
Yeah. Yeah, there's no reason for them to. I mean, it's horrible to say, but like I suppose in their eyes, the damage is already done and they are liability free. They don't have to do anything.
Yeah,
So
they're off acquiring cancer companies. That should scare the hell out of everybody.
yeah, yeah. And the other thing, yeah, exactly. That's this platform, the nucleoside probably modified RNA LNP platform specifically, which sometimes is being referred to as a plug and play system because you just have to swap out the spike gene and put in something else. I mean, they're experimenting with this and doing trials with this idea, for example, using flu genes all the time and in different contexts as well, like in this self-amplifying RNA stuff, which is even scarier. So my feeling is that this is going to become the modus operandi for all quote unquote vaccines. I really do mean quote unquote vaccines in the future. So it's and it makes sense for these companies to do this because they want to do this in industrial scale. But again, like there's nothing we can really do about it, and that's sad because there is no liability, and they are going to try and do things as cheaply as they can and make as much money as they can as well. Um, but the onus is really, I mean, regardless of whether or not they have liability, they have some kind of onus to ensure safe products. But it's crazy that I have to say that with so much doubt because it almost seems like they don't care because they don't have to because they won't suffer. Um, but if they do, I mean, I personally, I think they're very aware of everything that we're doing, especially what Kevin is doing. And if there are people who care about perhaps the eventuality of being sued, even as individuals, they will do something about this particular problem in the future. Because, like I said, I think this is going to become like, the way that they do everything in the future, the gene-based therapy type thing. And even for cancer, I mean, like Kevin said, they're buying cancer companies and they're like years ago, I wrote an article on the new mRNA cancer therapy. Can't remember the name off the top of my head. It was like, I don't know, 4 digits like the spike. I don't
Yeah,
remember what
they,
it
they
is.
purchased Seagen for $43 billion a few years ago and Trillium Health. And this is after the pandemic and after they saw their trial data, they went out and purchased cancer companies. So I think they see the signal. It's not hard to miss. And Waffick's paper goes through those various signals that you can find in databases and in just the literature. If you comb through all the literature, you can see that there is hyper-progressive tumor formation occurring, there's relapse occurring, and it's happening in younger people. And that should all be a sign that something has changed. There's a new toxin in the population, and that is when you see the age demographic shift with cancer, that should be alarming.
Exactly. Yeah. So it's not just the rare aggressive cancers, the reemergence after remission. It is the shift in age. Like more younger people who like really shouldn't be having the kinds of cancers that they're having are reporting getting them. And this is just what we are hearing about, by the way. It's always a tip of the iceberg situation.
It's
Um,
something, unfortunately, that the virus doesn't capture very well because it
yeah,
tends to only report things that were proximal to the vaccination date. And cancer is one of these things that probably takes a bit more time to show up than pulmonary embolism, strokes, and acute renal failure or myocarditis, right? Those are signals that are really acute and immediate, and you can tie them temporally to a vaccine incident, but cancer might happen years later, and so no one reports them into VAERS, or very few do. So it's a hidden signal.
really download it, read it. Even if certain people go to retract it, download it, read it. um, and yeah, that's about it for now.